Okay, so the second part of the question, on the diagram of the cell, label a partially permeable membrane. Use a ruler to draw the labeling line. So this is a partially permeable membrane. I'll zoom it for you guys. So this is where the partially permeable membrane is supposed to be. So you have to label it over here. Not this one, but this one over here. Right. Okay. So the concentration of this cytoplasm and the cell sap inside the root hair cell is greater than the concentration of the water in the soil around the root hair cell. Use your knowledge of osmosis to explain how water is absorbed into the root hair cell. So you know that in osmosis, the water travels from a region of a higher water potential to a region of lower water potential. So similarly over here, you can explain water molecules move randomly. They don't move in any specific direction. There is a greater concentration of them outside the cell than inside. So more of the water molecules will move into the cell, into the root hair cell, than out of it. Because plants need water, they will not give out water. It doesn't make sense, right? And they will absorb in the water through the partially permeable membrane. The solutes in the cell cannot go out through the partially permeable membrane. This one is like one type of answer you can explain. Another way of answering this question, you can just mention about how water molecules move from a region of high water potential to a region of low water potential. That is also correct. So you can either answer in this way or this way. You know, this question is actually really hard. Like, you'll be like, what? I don't even know what this cell is, what this root hair cell is. How am I supposed to answer this? So they actually gave you one hint over here, which means you have to use the idea of osmosis about the water molecules moving from a high water potential. So you know, for osmosis, that one key term you have to remember is about using the term water potential. This is very important because they changed the definition of it in the syllabus. Like, they didn't really change it, they just changed the term. It's not like using just region of high water potential from a region of high concentration, but now it's a term of using a region of higher water potential. So since it's actually the new definition, it's better, it's easy to remember, as osmosis is entirely to do with water, so it's easy for you to understand. So it's going to be high water potential. You can either mention, since they're talking about the concentration of cytoplasm, the cell sap is greater than concentration, and now they want how water is absorbed. You can explain the ideology of the osmosis and mentioning of the partially permeable membrane, and you can actually use the same ideology over here since there's a greater concentration. So it's going to be the same thing. You can rephrase it actually, and then mention about the water molecules moving randomly, or you can just state about high water potential to a low water potential. Since the definition up above here, they said that it's a diffusion of water molecules, so you can actually literally mention this like in this thing actually, and that you have to use that knowledge of that roots, like they will absorb the thing, they'll absorb the nutrients. Water is one of the nutrients that a plant requires, so they will not give that out. So why? So now part D. Root hair cells are tiny and there are hundreds of them on each plant root, such as how this helps to increase the rate at which the plants can take up water. So this is the features of root hairs. So they provide a large surface area, so more water can pass across the surface at any one time. So large surface area mean they're actually very thin. It doesn't mean that they're thick. It's just that a large surface area mean there are more molecules of water and other molecules that come in contact with the root hair cells. So that's why it's since there are hundreds of them, meaning there'll be more. The molecules will be more in contact with the root hair cells, so it provides a larger surface area when there are hundreds of them, so they can actually easily pass across the membrane anytime. So the main point you have to mention, it provides large surface area, actually, that's all. So now exercise 3.3, it's about osmosis in potatoes. So in this exercise, you'll practice drawing a results chart and recording numerical results. You will also construct a graph and evaluate the results. Question D is a good test of understanding your osmosis and your ability to use your knowledge in a new situation. Right. A student investigated the effect of different concentration of sugar solutions on some potato cylinders. Potato cylinders mean, potatoes, they're just cut in cylindrical shape, nothing special about them. He took a large potato and used a cork borer to cut out several cylinders, each exactly the same diameter. He trimmed the peel off the ends of the cylinders and then cut them into exactly 1 centimeter lengths. He then measured the mass of each piece. He placed one piece of potato in each of the 6 beakers. He then covered each piece with either water or one of the 5 different concentrations of sugar solutions. So either water or different concentration of sugar solutions. He used the same volume of solution in each beaker. Okay, he kept this thing constant. The student left the potato pieces in the beakers for 30 minutes. Then he removed them from the beakers, locked them dry with filter paper, and measured the mass again. These were the results he wrote down. So before piece A, 5 gram, 5 gram, almost 5, 4.9. And then solutions. So solution A, piece A was in distilled water. Piece B was in 0.1% of sugar solution. Piece C 0.2% sugar solution, 0.5% sugar solution. So the concentration increased. And after, so piece A before 5.2, after placing it in distilled water, it increased to 5.5. Piece B before 5.1 gram, after placing it in 0.1% sugar solution, it slightly increased to 5.2 gram. Piece C before it was placed in the solution, it was 4.9 gram, and after placing it, it remained the same, no difference. Piece D 5 gram. After placing it in 0.5% solution, it slightly increased by 0.3 grams. Piece E 5.1 grams reduced to 5 grams after placing it in 0.8% solution, and piece F 5.2 gram. After placing 1% solution, it decreased. So in the space below, draw a results table and fill in the student results. Include a row or column showing the change in mass. Take care to head each column and row fully with units. So basically, the first column that you have to show is the percentage concentration of the solution, and the first column that you mentioned is the pieces, like what are the name of the pieces, or you can say you can mention name of pieces, and then piece A, piece C, piece D, percentage of concentration, and then you can mention about the change in mass. So one example over here I could show you is this. This is one example. So you can mention about like this: percentage concentration of solution 0.0. Before soaking 5.2 gram. After soaking 5.5. Change is 0.3. So this is one sample of how you could draw. It's better if you actually state which piece was what. It's much more easier to understand, but this is also, it doesn't matter. And always mention the units. For the percentage concentration, it's better to include the percentage mark over here. They didn't include it over here, but it's best to include it as well. Right, so this is how you can draw a table. You have to basically use the results derived from here and include like this. It's not necessary to make it like this. You can make it your own style, because they want all these information to be in the table. So it can be either way, and the only thing that you have to additionally show is the change in mass of before and after. And obviously you have to label the table, and the drawing of the table is always in paper 6. It's never in paper 4. So it's usually about like 3 to 4 marks, so it's a lot actually. So you have to master how to draw. Actually, this information, when you will see like, oh my god, how do you like place in table, it's kind of of complicated. You know, this level of difficulty, it's never usually present in the paper 6 of the actual IGCSE question. Most of the questions in this workbook, they're actually of a higher level of difficulty compared to IGCSE papers, which is actually good because it kind of trains you to answer the less difficult questions in your real exam or your real IGCSE exams. So it's a good training. They won't really they won't really complicate your minds in this way, because I've seen a lot of questions about the drawing of table, but this one is exceptionally kind of really difficult, but difficult in the sense that you have to organize which one will go where, which one will go there actually, and it takes like probably 10 to 15 minutes because you have to include all of this, so it needs to be well organized. And yeah, for drawing a table, always remember you draw the table with a pencil, but you fill in the data with pen. This is how you're supposed to do it. You draw your table, like the rows and everything, like all this rows, you all draw in pencil, but the filling in of data you have to use with pen. Yeah. So right. Now decide if there are any anomalous results. If you think there are, draw a ring around them. So if you draw the table, you will see that it's going to be 0.5%. Why is it like, why is 0.5%? Right, so if you see the table over here, as the concentration of this solution increases, the change in mass decreases. Like 0% was 0.3 change. 0.1 percentage concentration of solution was 0.1 gram, 0.20, 0.5, suddenly it goes 0.3, but 0.8 and 0.1 actually decreases. So you can actually deduce that the anomalous result is the 0.5% concentration. So you can just draw a ring around or mention it somewhere. Yeah. Display the results as a line graph on the grid below. Put concentration of solution on the x-axis and change in mass on the y-axis. Remember to include units in your axis label. So I give you one tip. Normally in paper 6 they won't ever mention which labeling will go on which axis, which labeling will go on what axis. So you have to know that on the x-axis it's always the independent variable, the variable that is always changing, the one that you are changing, and the one that's on the y-axis is the dependent variable, the one that gets affected as a result of the independent variable. So if you see over here, this is the independent variable, the one that's constantly changing, and the one, the dependent variable that gets affected as a result of the independent variable. This is the dependent variable, change in mass. So this is one way how you could draw it. I actually, well, it's not fitting over here, but let me show you how I could do it. So as you can see over here, you actually need to draw the four grid, like the 4 types. Like, you know, this is the positive, this is the negative, this is the negative, and this is the positive. Like, you have to draw that 4 type of grid, not just like one, just not the 2. You have to draw like the 4, like, you know, positive, negative, like that type of grid. Why? Because you have the negative results, you have to plot them somewhere. So obviously you have to use this grid. So when plotting, they actually mentioned the question over here, a line graph. So when they mention about line graph, it can either be a best fit line, or you can plot it like this. I recommend you to plot it like this. Normally for paper 6, they always want you to draw a best fit line. They usually mention, but sometimes in some questions they don't mention. That means you have the freedom to choose either to connect all the points, or to draw a best fit line. So now when you're plotting the points, or when you're drawing best fit line, if there's a question asking about anomalous like, you know, identifying anomalous results before drawing the graph, then when you're drawing the graph, you do not touch the anomalous result. You just leave it there. You do not connect the points. You do not draw the best fit line over it. You just leave it like that, because that's not an accurate that's not an accurate one. That's not an accurate answer. So that's, you just leave it there. So you just connect the rest one over here, because this is the anomalous one, so you don't touch that. So you just connect it like this. And obviously when you are plotting, make sure your heading, there's a unit mention. Actually in the question, since they didn't mention like the percentage, so you don't really have to mention, but it's best to mention since it's about percentage concentration, so you can also mention it as well. And remember, when if you see over here, where was I? If you see over here the percentage concentration in the original question, they're all in one decimal places. That means your graph must be it must be drawn, all your points must be in one decimal places. Even the 0 must be 0.0 and 1.0 as well. So this is how it's supposed to be. I don't have a graph paper, so I did on an A4 paper, but for you guys, you must draw on a graph paper since the graph is already given to you, so it's good. So now I'll explain the next part of it in the next part. So yeah.